Hi, everybody. I am so excited for today's talk. This is our first non-biomedical engineering session, but still completely relevant and super interesting. We've got Tori Quintana. Tori, can you introduce yourself?
Yeah, hi, thank you so much for having me. I'm Tori. I'm currently a second year PhD student in the cancer biology program. And I'm so excited to be a part of this YouTube channel. So thank you, Lane, for inviting me.
Oh, this is so fun. Tori and I actually met at the beginning of both of our programs, right? Almost 2 years ago now.
Yeah, I know it's crazy how fast time flies.
Oh my gosh, so amazing. Okay, so I'm not in your program. I don't know a lot about it. Before we get into the research, can you just tell me a little bit about your lab, just a general overview about what it is that you do?
Yeah, so first I'll talk about like the program itself. So cancer biology is a really great, interesting program. I'm putting program in quotation marks because it doesn't have its own department, and it kind of makes up a really good mixture of different studies. So we study cancer, but there's a lot of people who study different aspects of cancer, and my lab is a great example of that. So my lab, we study a very basic science of understanding how DNA gets prepared by DNA damage. And that can be listed in many different types of diseases. It also just happens in everyday life. So what I study is how DNA damage can occur and accumulate and progress into cancer. So one of those pathways in which DNA repairs itself is the base excision repair pathway. And there are many different pathways where DNA gets repaired. But this pathway in particular repairs DNA after oxidation or alkylation or deamination damage. And the best example of that is by UV light. When you go outside and you get sunburnt, a lot of the damage that occurs is through oxidation damage. And so there are many different proteins that are part of this pathway. And my research looks at one of those specific proteins and how aberrant repair or how mutations in that protein can lead to even more mutations and lead to the progression of cancer.
That's crazy. It sounds like it's such a huge field with so many different aspects. Is everything in your program relating back to cancer and aging?
Most of it does, yes. So, and of course, there's also many different programs that have cancer within them as well, or that study cancer as well. But cancer biology in particular, most of the labs study cancer.
Okay, and so I know that you had a review paper out, and that was kind of what I was using a bit of background research for this. So interesting. I had to make a giant list of words to look up, had to, refresh back to my limited knowledge of genes and everything. This paper sounds so interesting. What were you doing for it?
Yeah, so I was so lucky that when the pandemic hit, my PI actually asked me in a postdoc to write this graphical review paper. And it was perfect timing because I was just starting to understand my thesis project. And it really focuses on the one protein I was talking about, and it's called NTHL1, and it's a DNA glycosylase. And our lab really loves this protein, along with some others. I'm biased, but we wrote this graphical review, and it's mostly, graphics, but we did talk about many of the aspects of this protein and how it's relevant within cancer, and in particular, this really rare multi-cancer syndrome that kind of encompasses many different types of cancer. So that's colorectal cancer, endometrial cancer, skin cancer, breast cancer. And so it's really interesting to see how, mutations in this one particular protein can lead to such rare and impactful diseases.
So I don't know if this was one of the key points, but I thought it was really interesting in here how you were talking about this NTHL1 is in a lot of different life forms, but it's a little bit different in humans, even compared to mice.
Yeah,
That's crazy.
it is. It's so I won't go into too much detail.
Yeah, not the focus, but
but yeah, the human NTHL1 is really interesting. Like I said, I won't go into too much detail, but the human NTHL1 homolog has this N-terminal tail, and the mouse does as well, but the human protein has this mitochondrial and nuclear localization site, in which it can be both in the mitochondria and in the nucleus. But it's really not well studied. So this N-terminal tail is really disordered. So we don't know if it interacts with other proteins or what it really does. And a lot of the mutations that we study in our lab lie within that region. So it's really interesting, finding all this data and trying to discover like, what is this protein really doing? Or how are these mutations diverting this protein from doing its actual job? And so that's kind of what me and one of the other postdocs in our lab are trying to understand. And it's really interesting. And I'm glad you picked up on that.
I love looking for the stuff. I love the little fun facts, that what gets me engaged. And then I also always look for like the bits that say we have no idea what's happening. And yeah, it looks like this one was just saying like there needs to be more research, basically.
Yeah, absolutely. And that's kind of a lot of what research is, right? Is we're just trying to find more of the facts and verify that things are really happening, and then all of a sudden things change and you thought it was going one direction and it's actually going the complete opposite way.
Totally. Oh yeah, that's research for sure. So okay, I understand NTHL1 a little bit, but can you give us a rundown of what it is? It's a glycolysis. That's an enzyme, right?
Yes.
This is so embarrassing.
No,
Glycosylase.
So I'm just going to give like the briefest understanding of the pathway, and then you'll understand what a glycosylase kind of does. So the base excision repair pathway is just one of the many pathways that repair DNA after damage or DNA damage.
Base excision repair pathway.
Right.
Got it. Okay.
And so I'll focus mostly on the long pathway. There's also a short pathway. But within the base excision repair pathway, there's two forms of glycosylases. There's monofunctional and bifunctional glycosylases. They kind of do what the name says. Monofunctional means that they can only remove the lesion off of the base. And they don't have lyase or glycosylase activity, and I'll get into that in a second. Now, there's bifunctional glycosylases that are able to remove the base, be able to cut the backbone of the DNA and create like a single-stranded break. So I'll mostly focus on that. And NTHL1 is one of the 4 or 5 glycosylases that can do that. When the glycosylase removes the base and cuts the single-stranded break, other different repair proteins come in that can anneal it. Polymerase beta can come in and put in the correct nucleotide, and then like different ligases come in and they can seal the nick and it can go on to continue replication. So all a bifunctional glycosylase or glycosylase does is remove the lesion off of the base. And there's many different types of glycosylases, and they're all specific to certain types of damage. So NTHL1 in specific can remove oxidative damage. Yeah.
And that's, for example, from sun exposure?
Yeah, so like one of those could be from sun exposure. Yeah.
So then what exactly are you doing in your lab? Are you what is an experiment that you do?
Yeah, so one of the different mutations within NTHL1 is known to lead to an accumulation of genomic instability, as well as an increase in incretin-dependent growth, which just means that it can move from like a normal state into a cancerous type state. And so what I'm doing right now is using a mouse model to understand how this mutation could lead to the progression of cancer under certain really stressful conditions. So one of the ways I'm testing this is by giving the mice an inflammatory agent to mimic chronic inflammation. So it's AOM DSS model, and it's really well known within the field. And so what I'm testing is to see if people, well, if mice who have this mutation are more susceptible to develop cancer than those with just wild type or just a normal NTHL1.
Okay. Okay, interesting. How do you have to do, how do you know if it's happened or not happened? Is that a PCR test, or is it culturing, or?
Like, what do you mean?
I don't even know. I'm not even sure. I just, okay, what I'm referencing is on Instagram, you always have these cute little videos of you doing experiments. I want to know what those are.
Oh, so yeah, so that's just one experiment. So most of like my wet lab experiments have to do with, let's see, like for cell culture example, we test for genomic instability. And one of those ways we can test for that is by immunofluorescence. We can look for the staining of micronuclei. And micronuclei are just cytosolic DNA that kind of get released from the cell when they're going under mitosis, and it's just a sign of genomic instability. So we can test that by staining the cells for DAPI, or a nuclear stain, and we can go under the microscope and we can see if there's cytosolic DNA, which can indicate that possibly something's going on or something's going wrong within these cells.
Oh, yes. Okay, that's exactly, yeah, that was exactly what I was wondering. I love immunofluorescence staining. I just think it's so pretty. It's one of my favorite things.
Yeah, it's really, I always thought it was like I should never say this, but I thought it was one of like a really straightforward experiments, but it can be pretty difficult when you're under the microscope and you're there for like 6 hours trying to get something, a really good photo, but it's definitely worth it at the end.
Ooh, okay, that actually leads me to my next questions. Something I've really been interested in is, I feel like a big part of research, we're always talking up the results, showing what the good side of stuff is. But I think a huge part of it, especially when you're an undergrad coming into the area, is the difficulties and like how long experiments take, or how many different ways an experiment can go wrong, and just general gaps in the area. What are your, I don't know, what are your main limitations and pet peeves with all this stuff?
Yeah. So it's kind of funny. That's the one thing in grad school that no one really prepares you for, is how much time and how much troubleshooting you actually do when creating and designing and doing experiments. And it's something that I've really struggled with. And I try, my best not to let it get in the way of like my progression of things or my outlook on just like normal life, but can definitely be challenging. And I think one of the biggest pet peeves that I have with myself is when like I do a really simple mistake that kind of just like leads the way for like a downhill progression from there. So like one example is like I mislabel a tube or something, or I don't label a tube or something, or I completely forget about adding something into like a master mix or something like that. And it's little things like that that I get on my own nerves that I do that kind of just make me have to redo this experiment again, or mess up the results or something like that.
Oh, I get that. The other day, I have to make these particle conjugations. It's a 6-hour thing. I was on the very last step, and then I was just feeling a little bit of a perfectionist mode. So I wanted to just retouch it a little bit, and I accidentally added it to the wrong tube and messed the entire thing up.
Oh no, that's a 6-hour experiment. Oh, you poor thing.
It happens.
Yeah, exactly. That's the outlook you have to have. It's just, it happens. I'll have to do it again. Just move on. But
yeah, definitely. Okay, well, it's nice to hear that that's actually, I was so embarrassed when I did it, but then I've told a couple of people and everybody's like, oh, that happens to me. So.
Yeah, exactly. Yeah, it's reminding yourself that you're human, and more than likely it happens to everybody.
Yeah. So I mentioned the Instagram, but I love your science Instagram page. So I want you to tell us a bit about that. How do you manage like sharing your research online, and kind of, because you do a lot of education, you do a lot of lifestyle, it's such a mixture.
Yeah. So I actually started my Instagram back in undergrad when I was applying to grad school, and I wanted a way to showcase how grad school really is. And at the time, I was really enthusiastic about grad school. I was so excited. And I just wanted to share my journey of like what it was like to go through rotations, what it was like to move to a new state and really not know anybody, or what it was like to pick a lab and to go through all these like new classes and try to balance, your life and research and school. And so it's led me to some like great friendships. Like that's almost how we met. And then just to be able to share my journey with like my family and friends and meet new people. I think, I struggle a lot of times because you get in this mindset of comparing yourself to a lot of other wonderful, incredible scientists out there who may be publishing a bit more than you, or they seem to be progressing in the lab faster than you. And I think, at times I have to remind myself that we're all in our own journey of our graduate school, whether you're in a master's or PhD program. And you can't, compare yourself to other people. And so I like to, be honest in my Instagram posts and be like, if I'm having a hard day, I just tell you, because I know more than likely a lot of other people are having like a hard week, a hard month, a hard year too. And it's just sometimes good to relate to people in that way rather than always showcasing, your best self. Because I know I definitely fall into the trap of doing that.
Yeah, I remember during kind of a couple months into quarantine last year, I remember you did a couple posts just about struggling and how you were managing it. And oh, I loved that because I was right there too. It was so nice to see, especially I think on these science Instagrams, there is a lot of these beautiful infographics where people are presenting stuff perfect. I love it, but it is a little intimidating sometimes.
Yeah. And it's like, kudos to them. I wish I was, very creative in the way I could, make these graphics, but I can't. But yeah, I think it's good to have like a mixture of both, right?
Right. It's a variety of styles. I think that some people really excel at that pristine. And then it's also so nice to have a little bit more of a real, this is life.
Yeah, exactly.
What do you do aside from research? And everything?
Yeah, so right now my biggest hobby is running, and I do that for many different reasons. One of them is just more of like to let go of all the stress that grad school and life comes with. It's one of the ways that I can get myself outside and be outside. In the lab, like, you're stuck in there for 9, 10, 12 hours a day, and sometimes you don't see the sun at all. So, running allows me to get outside at least for like an hour a day.
How, mention the marathon, please.
Right. So I actually just completed a marathon last Sunday. And that was really probably one of the most challenging things that I've done, juggling grad school and training. It was really, it took a lot of discipline and, really making sure that I was on game all the time. And I think it helped me in the long term understand like what I'm capable of. And if I can handle really, really hard times, I can handle a lot of other really hard things too. So it was a really big and crazy accomplishment, and I'm definitely resting now. But yeah, that was like a lot of fun.
I can't believe it. It's so impressive. A marathon. And it was just by yourself, or it was like through an online sort of, was it an online program or was it just?
Well, it was supposed to be like an in-person race, but of course, things happened and it got canceled, but I was still training, so I figured I'd still do it. But yeah, so I ran it all by myself. I ran from one side of Tucson to the other side of Tucson. And my headphones died like the moment I started. So I listened to absolutely nothing the whole 3 hours and some minutes.
How was that? Boring?
It actually wasn't that bad. I just replayed like 4 songs in my head the whole time.
I can't believe you did it so fast also. That's pretty, are you gonna do any competitions once in-person things come back up?
I don't know. I might. I think it would be fun. Right now, I think the biggest thing is just, staying in shape and just being healthy and letting my body recover a little bit after
Mm.
such like a big training cycle. But I think it would be fun once things get back to normal to maybe do a couple races and just be with people and running again.
Oh, I love it. Okay, I know not all of this is science related, but I think that it's such a relevant topic. We're not just in the lab doing experiments all day.
Yeah. Yeah, exactly. Yeah, as much as I love science and I love my research, it's so important to have other hobbies and, have a life outside of research. And, you know, I definitely get caught in the trap of like constantly thinking about work in my lab and what I have to do next. But it's really good to let it go and, enjoy life. We're so young still. It's good to, you know, just have other hobbies out there.
Oh, completely. So moving forward, what's your next, how long is this program that you're in right now? What's the general, do you have your theme of um, dissertation?
Yeah. So of course, years vary depending on how much I have left. But the average is around 5 or 6. So I have about 3 more years, and I'm coming up at the end of my second year. So I'll be taking my comp exam. It seems that my comp exam is pretty late in the game in comparison to other departments or programs. So I'm a little nervous for that, but I do have the theme of my proposal or my thesis, and that'll be on NTHL1, looking more at that mouse project that I was talking about a bit earlier, which is really exciting. It's going to be really busy, but it's also really exciting to see myself, come from, and I bet you could see this too, from where you started, a little, less than 2 years ago, not knowing what you're going to do. And then to now, you passed your comp exam. So being a PhD candidate and, progressing so fast, it's just amazing to see the progression of things.
It's fun. It's definitely fun to look back and get a little perspective on the progress.
Mm-hmm.
What do you have to do for your comp exam? Is that a test or is it oral or?
Yeah, so we have like a mixture of both. I really, really like the way the cancer biology program sets it up. So we write an F31, and it's like um, you don't have to send it to the NIH, but you have to have an F31 style proposal written. And you actually take a class in your second year with how to write that F31. So it's really, really nicely set up. Then you send that to your committee, they look over it, and they can either accept it or accept it with revisions, or they fail. But usually it's always accepted or accepted with revisions. Once that's done, a month later, you take your oral exam, and that oral exam is almost a test on that F31 proposal, mixed in with some just general knowledge with the classes that you took. From what I've known, it can last anywhere from 1 to 3 hours. But I like the style. I think, if you write your F31, that gives you a really good standing point on what you have to study, what you already know, and then that month before your oral exam, you just kind of refresh and more on the general knowledge that you have from your classes.
Oh, I think that's quite similar to BME. I did my qualifying exam, but I haven't done comps yet. That'll
Oh,
probably be
okay.
next semester. Yeah. So you got to tell me how it goes.
Oh my gosh. Yeah, I definitely will. What's the difference between your qualifying and your comp exam?
So for the BME program at the U of A, the qualifying exam is based on courses. Like there's a certain
Okay.
set of courses that we have to take. And that was just a big exam. It was a full day thing, one for each class that we had to take. A couple of them were electives. That was so hard. And that was actually right during the pandemic too. And it was the first time, so it was really nice because it was the first time we got to see people in person after like
Yeah.
such a long time. But comps exam used to be similar. It was also coursework, but now they've changed just this year to a format pretty much exactly
Okay.
like what you're saying, I think. I'm not sure what type of
Yeah.
grant it is, but
Yeah. I think that way it's like it sets you up. So if you want to submit it, I know there's different, funding sources. There's like the NSF, the F31s, there's so many different ones, but I think it really sets you up. Like if you have a substantial amount of preliminary data, you can send it and try to get a grant. As a third or second year student, I think it's really, really great.
Yeah, absolutely. Especially if you're interested in going into academia. I mean, grant writing is such a big part of that.
Yeah, exactly. And it's so difficult. It's so difficult. I've struggled so hard with, changing the way I've written for, so many years into, really deep scientific writing. So it's been challenging, but it's really, it's a good test to see what you can do.
Are you doing it on the NTHL1?
Mm-hmm.
Wow, that's awesome. Oh, that's okay. I cannot wait to see future papers that you churn out.
Oh, thank you so much. I'm excited for you too. It's been so amazing seeing you progress in your program. I love your YouTube channel. I love like your now Instagram and how you've been sharing so much of your lab now. It's so exciting. And it's amazing to see so many different aspects of research. At times I get so caught up in just my own lab and how diverse my own lab is, but seeing it from, so many different perspectives, it's just so amazing.
I couldn't agree more. It's so fun to see and talk to people from different labs because I feel like we have a very similar experience in a lot of ways. But then also just the research. I mean, everybody is in their little niche and it's so fascinating. I don't think we realize. I always feel like a bit of an idiot because I just know how much I
Yeah.
don't know. But then you talk to people and you're like, okay, I don't know a lot, but I do know a little bit about my tiny thing.
Right.
Yeah, well, I don't know where this YouTube thing is going to go, but I'm just using it as an excuse right now to talk to people and learn stuff.
Hey, you
Which,
know what? That's the best way to do it. And hopefully, things will get better soon and these conversations can move more in person and your YouTube channel will just like grow and flourish.
oh my gosh, we got to do one in person for sure. We can do a little tour of your lab maybe.
That would be great. Yeah. My lab is getting so big now. Let me see, we have 4 PhD students now.
Whoa.
Yeah, I used to be the only one. So now we have 4 and including me. And I'm so happy.
Oh, that's awesome. Do you guys have undergrad students or volunteers as well?
Yeah, so we have 3 undergrads right now. And then we have 5 postdocs.
Okay. Oh, wow. Wow.
And then we have 1 research scientist, a tech, and a lab manager.
Oh my
We
gosh.
have a very big lab.
Do you have your lab meetings all together? Do you get to learn about each other's research or is it quite separate?
Yeah, so we have lab meeting almost every single week. And it used to be where we'd all, get in a really big conference room and we'd discuss things and it was really great. And of course now it's over Zoom and we still discuss, but it's a little different. But yeah, our research goes every direction. We have people working, in biochemistry, we have people working with animal models, we have cell culture work, we have people dealing with cancers, some people dealing with like lupus, or some people dealing with gastric cancer, and some people not even dealing with cancer, just more basic biology type stuff. So it's really amazing to see how diverse our lab is. I'm constantly learning new things, but it's really exciting.
Wow, that is such a good opportunity to learn about so many different areas of the same general field.
Yeah. Like you said,
Oh,
it's like you constantly think about like like you're learning things that you didn't know. So I feel really grateful that I have such like a strong lab in which they can help and teach me so many different things.
it's a cool opportunity to rub elbows with all these cool people.
Yeah, exactly.
Oh, this has been awesome. I'm so happy you came on.
No,
And I'm
thank you for inviting me.
oh, of course. I'm really excited for this in-person one we might do in the future.
Absolutely.
Okay, awesome. I will talk to you later.